So, hello everyone. Welcome to the microbes live stream. I'm a bit nervous, obviously, since I've streamed only once. I hope my frame rate is good enough because I'm using my Canon DSLR and I'm not really sure if um, what I'm seeing in OBS is a bit choppy. So I'm not really sure, but welcome. Today I will be introducing you to microscopy. It's one of my favorite subjects, and I have gotten my microscope like 4 years ago. It's insane. I've used it so much. It's an introductory, not necessarily introductory microscope. And but it's good enough for the job and it produces decent optical quality. Let's begin. Today I have prepared. I've gone and sampled um, from like forgot. I've gone and sampled and brought a sample from freshwater. Well, I wouldn't call this fresh because it's like from wastewater. Um, but it's, I would say, perfect for for the job. And it's smelly though. No one talks about how smelly microscopy is. Um, so let's begin. Let me change the angle so you can see what I'm doing a bit better. Come on, camera, please focus. Okay, for some reason my DSLR has decided to stop working. Um, Perfectly. Okay, let's begin properly. Okay, so preparing the slide, I have a few tools that I need. It's a pipette. It's a plastic one. It's like I got a bag of them, like €2 or €3. It's insane how cheap they are. And my trusty tweezers. I also got a set of them. Um, Also pretty cheap. You don't need the expensive equipment, especially in like those disposables. This is definitely disposables. We have our slide. It's a very useful tool. Well, it's a necessary tool for microscopy because you obviously need some way to mount um, your slide, right? So let's begin. So first of all, you need to um, open the sample. You know, I've tightly capped it because any leaks would be, well, not devastating, but they would be bad. You know, um, we need just a tiny drop of water with hopefully sediment. As you can see here, there's gunk inside of the pipette. It's a bit too much, but as I said before, we do not need much, like we need half a drop essentially. And then we need to cap it off with our cover slip. Cover slips are necessary because it makes your sample flat, first of all, and secondly, it allows your objective not to touch your sample. I'm going to press the sample a bit so it gets as flat as possible. A bubble of air has formed inside. I do not know if you can see it. Let me. There we go. You see? This big bubble in the right next to the splotch of gunk. That doesn't really matter since it's going to be filled probably anyways. Let me focus again. Thank you. I'm glad you think it's cool. Now we're going to mount the slide into the microscope. Oh, let me change the view so you can see what I'm doing. There we go. So, hello, you can see my hand right here. This is the slide, and I'm going to dismount the previous slide. I had this is like a sample slide from a toy microscope that I use mainly to calibrate. And here's our slide. And we are very lucky since we can already see stuff. Stuff is good in general. Let me bring the microscope a bit closer to me. Just cameras. I'm going to cap off the sample since we do not want to, we do not want to throw it away or disturb it, because if it spills, massive problem. Well, not massive problem, but So, let me readjust a bit my main camera. There's a cable in the way, but it's okay. So, as you can see, we are on the sample. We can see a few stuff. The one microbe that I actually saw has bolted somewhere, but we're gonna find it. We're definitely gonna find it. Microscopes have a few different lenses that we're going to use today. First off, it's the 4 times magnification lens. Those are the objective lenses, Let me magnify a bit so you can see better what I'm showing. Right. This is the 4x magnification. This is the 10x magnification. This is the 40x magnification. And on the other side, I think you can see in the other camera, it's the 100x magnification. Those are all important, but the 100x magnification is an oil objective, and we need some immersion oil for it to work properly. I can show you in a bit what I mean. Because it has to do with the refractive index of the air and the oil. So, let me bring it back down so you can see properly what I want you to see. Let me adjust the camera. There we go. So you can see me talk as well. um, so let's begin. So in right in the middle, you can see a couple of blobs. Those blobs are definitely microbes, but I'm not sure what they are yet. They might be eggs also. You know, let me fix it a bit. Ah, right. Okay, perfectly. Um, those those are not microbes, but those moving are definitely microbes. They're pretty hard to follow because they move quite fast for their size and for what you would expect of a microorganism. There's another one, a bigger one. This is, I think, Paramecium. It's a species of ciliate, meaning they have some hair-like structures that they use for locomotion and for feeding. They feed mainly on bacteria, I think, I'm pretty sure, and either detritus, but very small particles. This is another species of ciliate. Let's change to the 40 times lens. It's pretty hard to focus sometimes. There it is. Those are some of the actually slowest moving. Oh, it's too dark. Let me adjust. Ah, a bit better. You can see some dirt and uh, um, you can see that the lenses are not quite as clean as I would like them to be. Let me check again for the brightness, because my phone that I'm using as a camera is not behaving as well as I would like it to. Anyways, and I need the aperture of the microscope to be quite closed in order to be able to see well. You know, on another stream, I'm probably going to do some introduction to the basic concept of microscopy, because it's quite fascinating and fun, I would say. You know. Um. So yeah. Let me change the lens so we can try and find some other easier to follow microorganism. Ah, so this is quite a fun microorganism. It's a microanimal, in fact. This is a worm. This I do not remember their names. Nematode. Yep, I remembered. And they live on. They're the most populous animals on Earth. You know, I think it counts on like hundreds of trillions of individuals, and definitely the highest biomass, uh, the highest animal biomass. Um, they're quite fun to watch around. They're quite motile, and they they eat everything. Let me find the focus again, because there we go. It's wiggling around like a worm. But there are some very diverse group of animals They're very, very small, but they can, some nematode species can grow up to centimeters long. They're also used in research a lot because um, they're used as model animals. And they're quite useful for that. You see some of its poop actually. This blob in the end of the animal. Those are all freshwater organisms. Uh, but nematodes can be found on land, in the soil, in fact. Um, in the sea, there are a lot of saltwater nematodes, a lot of species of them. Here's, I think that's another one, probably dead from something. Let me. Yeah, I think it's dead indeed. It's not moving at all. I do not know that much about them, but here you can see its digestive tract. You know, because that tube, tube-like organ inside of it. Um, it's the brighter part in the animal, right here. There, the other one, I think it's still there. But yeah, but it has found its way a bit. It's moved somewhat. I'm not sure what it's trying to do, but you know. So let's see what else we can find. This thing, the spiral shape, I have no idea what it is. Let me adjust the brightness. There we go. No idea. Let's zoom in a bit so we can take a closer look. And readjust. That's going to be a bit annoying, but I'm going to get used of it. So this could be some kind of bacteria. There are lots and lots of bacteria that have similar shape, but I cannot identify this as something um specific, you know. And there are other spiral shapes next to it, but I'm also not sure what those are about. So yeah, but they're quite cool actually. They're very cool. Let me open the aperture a bit. So there we go. Um, but yeah, I have no idea what those are. These though, this structure with the globular particles might be a sack of eggs because there are lots of animals, microanimals like rotifers or tardigrades, but tardigrades have different kind of eggs. So yeah, a comment says Pyrulium, but I'm not so sure. Not sure. So, but it could be. Let me search it up actually. Yeah, no, I don't think so. It's not green, and spirulina species are cyanobacteria, but this is definitely not cyanobacteria because it doesn't have the blue green the blue green color that they usually have. Spirulina, apparently, I didn't know that actually, is a substitute, a food substitute. But yeah, you live and learn, I guess. So let's change lenses once more. Adjust. And let's search for microbes. So, those two are some of the most famous ciliates. This is Paramecium species, probably Paramecium caudatum, a very common species of Paramecium. It's a type of ciliate, meaning again, as you can probably see around the microbe, there are those hair-like structures that uh, cycle water around them and cause particles to flow. On the left side of the left paramecium, you can actually see the current, the spiral current that they create in order to feed themselves. And those particles are uh, very small, small enough to fit in their mouth because they have a mouth. Um, Actually, well, many microbes have a mouth. Um, it's quite cool, and they work in several different ways. Like, obviously, in animals, it's quite common for the mouth to be a hinge that bites and strips leaves or chops down meat or fruit or this kind of stuff. But there are other kind of mouths. There are um, the worm mouths that work mostly like um, suction. They work with suction the water, the aquatic. Found the right word. The aquatic worms work like that. Many of them, at least. Some other work with have tongues or some other kind of organ that scrapes that works like a rasp and scrapes off flesh or um, detritus from the ground, buried leaves. Earthworms work like that, exactly like that. Um, so yeah, microbe mouths are way more diverse because um, they come from very different clades. Like um, while vertebrates are one singular clade, and the mouths are pretty common, like pretty similar, not pretty common, pretty similar between them. Um, in Ciliates, for example, which is a similar type of clade, they're very very diverse. There are other ciliates that these ciliates that use their mouths to form cyclones, to form spiral currents, you know, to force particulates, force particles into their oral cavities. Um, there are others that form different types of currents, like just form linear currents, like this paramecium. Um, most linear, but yeah, there are other species of microbes called Vorticella species that you can actually see two spiral patterns forming around it, well, above it, or in front of their mouths, in front of their oral cavities, anyways. So yeah, um, and it's quite important. It's quite interesting to observe them. So these paramecia also are also used as model organisms, not as widely as, for example, nematodes, but they are quite common, and they're the most used species, the most studied species, because the most studied genus, shall I say, because they're very, very common. Like in wastewater, they're very important in wastewater. Um, just everywhere. And there are also a lot of species um, of this ciliate. A lot of species in this genus. This is Paramecium caudatum. I'm pretty sure. I have a book here for identification, but I'm not gonna pull it out now. Maybe another time. Um, that we're gonna have a bit more time. This is a bit of an introduction to microscopy and to this channel. Um, so, as you can see, this microbe has a very common, very common in ciliates. Um, organelle. Organelles are like the equivalent of organs in multicellular organisms, but in unicellular organisms, which means a single cell and many cells accordingly, um, it has similar structures which perform different functions. For example, the mitochondrion, which, as many of you may know, is the powerhouse of the cell. Well, some in a way, it produces ATP, which is the energy molecule essentially. But for example, in the photosynthetic organisms, it's the energy comes from the sun. So you could say that the chloroplast is also a powerhouse, but it doesn't exactly produce the ATP. So whatever. Anyways, so in these cells you can see the cytoplasm. The cytoplasm is essentially the inside of the cell, the, well, let's say meat, but it's liquid. It's not like in multicellular organisms where everything has its place. The cytoplasm, let's focus on this one, which is moving a bit less and it's not making our lives that difficult. Anyways, you can see the particles inside moving, and that's essentially plays the role of, let me find those again. There we go. Of the blood. Well, the blood plays the role of nutrient and oxygen transportation. You know, so there we go. Better, I think. So it takes sugars and other nutrients and circulates them around the organism. Oh, it left us. There we go. Found another. So yeah, the cytoplasm essentially contains everything that is inside the cell: the organelles, the food, the globules, and um, cysts essentially that contain the food. The core, the core, the nucleus. Found the word. So yeah, you can see the particles moving, and this is essentially this. Everything plays a role. Well, most things. You can also see some globules on the outside. Now, it's a bit difficult for me to show you because they're quite small. Can you see the two? They're like cysts, like two spheres on, like, how can I explain it? On the upper side of the organism right now, that they open and they close. Those have a very specific function, and it is to keep water out of the microorganism, out of the organism, because there's a process, some of you may have heard of it, that's called osmosis. Osmosis essentially makes works around a division. For example, if you have a membrane, let's say it is the cell membrane which plays this role essentially. Let's say you have a membrane, and this membrane divides the outside of the cell and the inside of the cell. The outside of the cell has way more water than the inside of the cell. The inside of the cell has way more nutrients than the outside of the cell. So all of those nutrients want to go outside of the cell, you know, and all of this water wants to go inside of the cell. So while the cell membrane is impermeable to those nutrients, except on specific conditions, on some specific conditions, the water can actually move between them, between the outside and the inside. And because of this process, and because the membrane is permeable to water, well, not that permeable to water, but still, let me find something again. There we go. The water tries to go inside the cell. So the cell has those cysts, that those organelles that expel water out of the cell, and this helps the cell stay alive. So yeah, right now in the center of the screen you can see one. It works very quickly actually. It's essentially a water pump. Obviously, as you can see again the cilia around the cell, maybe you can see them a bit better, like hair-like structures. Um, that move a lot. They have specific patterns depending on which direction the cell wants to move, and um, whether it wants to eat or not. Because in this case, eating is interfering with movement, and this is very um, very important because the water that um, the oral cilia move um, is is a very big percentage of the water that the somatic cilia move. So if you eat and move with the same essentially organelles, if you eat at the same time as you move, you're gonna move in different directions. And also, they need to use their somatic cilia to counteract the oral cilia in order to stay still. Um, now, this is a different species of ciliate. And in this species, I'm pretty sure that this central. There it is. It's the same organelle. It's the water expelling one. It has a particular name, but I cannot remember it right now. But yeah, this also works quite often, quite frequently. And here you see it again, expelling water. Maybe we can see the formation of a food vacuole. Vacuole essentially is an enclosed space within the cell, which is enclosed by the same type of membrane as the cell membrane, but it's sort of a different structure and has less proteins also on the membrane. And this is a smaller ciliate and quite a fast moving one. I'm not sure about the species or genus. I'm not sure about the species or I'm pretty sure about the genus on this one. I'm gonna pull out the book. I cannot resist. So let me. For now, I have this system for the books, so you can see what I'm seeing, sort of. So, this is the general guide to identification, essentially, to ciliates. And it's like so many of them, so many of them, and I think those belong to the clade called Podea, this clade. And let me rotate a bit. Yeah, I think a bit better. Let me focus. There we go. Zoom in a bit more. I think you can see the similarities in shape there. Like there are so many criteria that you need to focus on, and you need to consider when identifying a microbe. But this this part, the oral cavity, is very distinct in this clade. Now, this is a bit of an old book, so I say clade because I'm not sure about the overall classification. And there are quite a lot of mistakes because gene sequencing has gotten into the game recently, and that helps with identification and with classification also. A lot, and it's very important to have to sequence any microbe before you um, definitively identify it, because well, there are some species like Paramecium that's disturbing the other ciliates here. That you can definitely identify. Like, I'm pretty sure this is I'm definitely sure that this is Paramecium species. I'm not so sure if it's Paramecium caudatum, but who cares in the end? Like, well, yeah. But in any case, I'm pretty sure that's it, because though as you can see here, there's again pretty similar form, and this is important to remember that many of those microbes could belong, like, have very similar body shapes and features, but they belong to vastly different clades, and that's annoying. But it is what it is, and it's very fun to try and identify microbe species. Let me do a simple search. There's the same shape, and actually, let me change back. This might actually be Colpidium, because I think you can see the oral cavity. Can you see the cone shape? This cone shape is the same cone shape here, and this is the only identifying feature. So yeah, it might be Colpidium. Colpidium colpoda, it says here, but I'm not trusting necessarily that this is that particular species. Let me go back. Yeah, it's a bit different than the actual Colpodia clade. I think it's a family, but still I'm not so sure. Anyways, that's about this Colpidium. Probably Colpidium again. Let me put the book on the side and let's continue our search. There we go. Fix the camera once more. And yeah, let's search some more. Also on the Paramecium, you can see this is the oral cavity, and right in the middle of the organism you can see, you could see the mouth. Loads of microbes here. Those smaller microbes, especially ciliates, are very difficult to identify because they have no discernible, no distinct features that you can say, ah, yeah, this is this species or this is that particular species. Oh, let me pick up the book again. So yeah, it's a bit difficult or practically impossible for some to identify. Some small species of ciliates have very distinct features, especially um, vortichellids or relatives of Vorticella, Epistelis or similar clades, similar genera. So yeah, um, let's see if we can find something else around here. Just for brightness, maybe a bit better. Yeah, there we go. And let's continue. Generally, so this is a problem that we will encounter very much in microscopy, is bubbles. So there is this massive bubble that has formed right here. Also, lots of paramecium. Yeah, the bubble is moving because the water is evaporating. There's like so little water inside of the slide, like it's a drop, and it's pressed so thin. So all around the sides, the water is constantly evaporating, and it's important to understand that all of this water evaporating concentrates everything else inside the sample. So we need to replenish that water. So I have right here a jug of distilled water, and we are going to replenish some of the water. So give me one moment to bring a small cup so we can draw some water, and I'll be right back. I will leave you with this paramecium over here. Let me zoom in a bit, and there we go, and the moving water. See you in a moment. So I am back, and let me fix the audio a bit. Okay, let's see. I think this is fine for now. Okay, so I see that the water has moved quite a bit. So let me change the view. Well, there we go. So we're gonna pour some water on the cup and place a tiny bit on the slide. So I need to be a bit careful because it's a small cup. There we go. I'm going to use a very small amount, and the rest is probably going to waste. But because I need to do a very specific thing, I need the water to be quite deep. So what I'm going to do is, let me focus where I should. There we go. So how we're going to put such little water inside the slide, and how are we going to put it inside the slide? So let me pull out the slide. Let me focus on the slide so you can see the bubble, how much it is grown. Come on. Yep. See, it's huge. So we need to replenish that water. So what we're going to do is, for some reason I need to focus manually, so I'm going to take my tweezers and dunk them inside the water and close them. So this has pulled a tiny amount of water. So I can now just place it on the slide and on the side of the cover slip and open it. It's going to take a bit of an effort, and there we go. Now, theoretically, the water is going to be replenished. But, yep. I hope I can show it to you. Right now, the slide is mostly water. Oh, this is worse. I probably need a black background for you to see it better. Maybe. Yep. Anyways, so now we can put the slide back. I generally do it on the microscope because it doesn't really matter, but right now I wanted to show you. So there you go. So let's go back to the microscope. Now there is a problem. We need to find everything again, because let me adjust. There we go. Because right now everything that we knew where it was mostly has been moved by the water that we just added. So let's search around and find out. Let me put this water around. Okay. For a paramecium again. A bubble that has been left. Some microplastics. Microplastics are very, very common everywhere. Like, I've never taken a sample out of anywhere and not seen such not seen microplastics. In this sample, you are not going to see many photosynthetic microorganisms because it's from wastewater. So yeah, I'm very curious as to what this is. I think it's bacteria, but I'm not really sure. Yep, it's bacteria. Let me readjust. Come on. There we go. It's a very large amount of bacteria. Let me open. Not sure why they're growing in such a shape, but it probably has to do with nutrient concentration. But yeah, pretty cool. Those strings, by the way, that you see, those very thin strings. By the way, sorry about the dirt on the microscope, but I'm going to clean it very soon, but until I clean it, you're gonna have to suffer with me because I hate it and I'm not sure where this dirt is, but I definitely need to clean it. So yeah, those strings, anyways, are filamentous bacteria. They're called filaments because it's a lot of bacteria that are essentially stringed together. They're stuck together and they form these long strings. Some of those may not be filamentous bacteria and may be entirely one single cell, but because it's very long, it can be. It's very long. It can take all the oxygen and all the nutrients it needs through osmosis because I'm truly not sure how bacteria feed because it's a bit it's not what I've read about. Bacteria is not are not what I've read about a lot. So yeah, I'm going to search it up and I'm going to tell you. By the way, I'm streaming at Tuesday and Friday at afternoon to evening. I'm not sure about the hours yet, but yeah, afternoon to evening. Eastern European Standard Time. So yeah. Anyways, back to searching. Come on. There we go. Once more. I think I see something interesting, so let's zoom in. So, I have a feeling that we are seeing something very, very amazing. Let me adjust the brightness because there we go. So I am pretty sure that this is an amoeba, and amoeba are also unicellular microorganisms, but they're different than ciliates, and the difference is that they do not have cilia. But also, amoeba are the most well-known amongst common people. Well, not common people, but amongst people who have no immersion in this subject. So amoeba are the flowy, let's say, microorganisms because their cytoplasm is the most it flows essentially, it arranges and it flows according to where they want to go, and they form those extensions that are called pseudopodia, and those pseudopodia are essentially extensions of their cytoplasm, and they use that to move to eat. They eat by engulfing stuff like let's say this is the amoeba, and it extends one pseudopodium, it extends another pseudopodium, and then they use those pseudopodia to engulf their prey. But this is a shelled amoeba. They're called testate amoeba, and essentially the test is the shell. I'm pretty sure it comes from Latin. But yeah, now this shell is vaguely globular, and it's quite large as well. It's very, very large. I've seen a fair amount of shelled amoeba before, but this one is quite huge. It's one of the biggest ones I've ever seen. They also use pseudopodia, but their pseudopodia are like very, very thin, usually because it's like strands, it's like also hairs, but way thicker than the cilia of the ciliates. So yeah, let's see if I can find them. Those around it are bacteria, and those are some ciliates, dead ciliates, that for some reason haven't had their cell membrane dissolved. You know, usually after death, it just breaks down because it cannot hold the osmotic pressure, and internal structure fails that keeps the membrane in shape to form the shape of the microorganism. But yeah, maybe we can find at some point a dying microbe and we can observe. Ah, yeah, this one. Right in the middle of the screen, right now, you can see the cell membrane breaking. Uh, you can see right next to this, a bit above, um, there's the uh, how it should look like, and on the side facing that microbe, the central one, the membrane has broken. Quite lucky, in fact. Let me switch to the 100 objective and see if we can see something. But I highly doubt it because it's quite. Yep. I may have ruined it. Anyways, uh-huh. Yeah, if you crash the objective, the lens onto the slide, everything is gonna go everywhere because you are pushing a very small amount of water around. So yeah, those are amoeba again, but I think we've lost the ciliates. Let's zoom in again and see what we can find. What else we can find around this amoeba? There's the one microbe, and those are dead microbes. Those are shells of diatoms. I told you you wouldn't see live ones, but those are dead. Yep. Maybe eaten and spit out because loads of microorganisms eat them. Here are the shells right in the center of the screen. Okay, better. So yeah, very impressive, very nice. Let me. I think now the sound is gonna be a bit better, a bit louder. Sorry if I've changed that, and but yeah, let's keep going. I use two methods to search. The first method is like to scan around with the 4x magnification lens, and the second one is just panning around with the 40 times, because sometimes there are small things that you miss with the 4x magnification, and they can be quite interesting, but many times also you just waste your time. And that's quite common. This is another filamentous bacterium, a filamentous string of bacteria, possibly. I think it's a string at least. But yeah. This long thing might have been a dead nematode. This bendy thing. But yeah, I'm not sure. More bacteria. Here you can see in this filament that there are small beads inside it. I think those are the individuals, but I'm not quite sure. I'm definitely not quite sure, but I may search it at some point, because but it's quite difficult to search for this, for bacteria, because there are so many species, way more than nematodes or ciliates or eukaryotic microbes. Eukaryotic means that they have a core, a nucleus. Bacteria do not have a nucleus, by the way. So yeah. Here is the bacterial growth, which is larger, I think. Not very sure though. It's very large though, still very impressive. And this, I think, again, the nematode. You cannot see it yet, but let me change the exposure again. There it is. There's the growth. It's very large. Very, very large. Bigger than I thought. But yeah. Let's see what else we can find. Or it may be a different growth. I think we found traffic. Again. Nematodes and Paramecia. This is a very large worm. Very large nematode, actually. This string is in the middle of the screen. Let me refocus again. There it is. This is a single filament, like it's huge. There it is. Moving also. Readjust, come on. It's annoying me. There you go. The quality is a bit worse, I'm sorry about that, but right now my phone is not cooperating. Well, the app is not cooperating, so yeah. Let me try it here. There we go. There, finally, I did it. There you can see it moving. And they move fairly slowly, I would say, but, you know, someone else may say fast, but they're definitely moving. Ah, the intruder, permission. Let's change back. Here we go. Right now we're in focus. I think I'm seeing some possibly even dead eggs. Those. I have no idea what they are. But they might be eggs of some kind. So yeah. No idea though. By the way, you're gonna hear me saying I have no idea a lot because I truly have no idea, but that's the thing. Microscopy is very, very strange and also very difficult. because you need to learn to track things with the microscope, which is a skill that I developed after like a year, and I'm still not perfect at it, or at least as good as I would like to, but you know, things are improving. At some point I would like to talk to you about sampling methods, but I'm not sure how I would do that, because I would need to be outside sampling, but we'll see. We'll see. I was hoping in this sample also for some Porticella, but I'm not seeing any, to be honest. I may switch to another slide, like make a new one, because this is full of paramecia and nothing else. Not that paramecia are not interesting. Don't get me wrong, I love them, but you know. This is a Colmer nematode. Let's zoom in. Maybe we can see stuff inside it. Yep. Nematodes have muscles, since they're animals, and I think I found its, well, let's say, excrement point. I think this is its digestive tract. Like, it begins like right here. I'm not sure if you can see that, but in the mouth there's a hole. I can see it, but I'm not sure if you can. And in the middle you can see the digestive tract, but at some point here it goes into a different way, like, because nematodes have, well, I'm not sure. Maybe it's this structure in the middle of the screen. It's genitals. Not sure. Quite possibly. Because I think, yep, it's well, I will say it, whatever. It's asshole. It's back here. I'm not very sure. It's moving a lot. Because there's another hole, I think, another opening in the skin here, and I'm pretty sure this is its asshole, because the digestive tract comes up to here. So yeah, I think those are the genitals. Guys, we found worm sex. Yep. This is a pebble, small pebble, rock, stone, whatever you want to call it. Yep. Right now you can see you've seen worm genitals. This one is fast. This worm. I am pretty sure this slide is empty otherwise. Like, this is the end of the slide, or this is the end of the slide. Ah, no, this is the end of the slide. This is the previous end of the slide before I changed the water. So yeah. I'm gonna do a quick scan and see if we can find anything else before we put another sample in. Because you never know what you're gonna find. Ah, see? That's what I'm talking about. So, this microbe, this little moving thing is, oh god, yep, there we go. This is a euplotis. It's a ciliate again, but it's a ciliate that has sort of legs. It literally walks. This is, I'm not sure what euplotis species is, but it's quite cool actually. You can see the little legs. It's essentially the cilia, but you know. You can see it working on the detritus. It's quite difficult to follow, especially from the screen, because of lag. There's another one. 2 of them. Those legs are essentially bundles of cilia that work together to create a structure called cirri. Cirri are sort of legs. Well, they work similar to legs. They step on things and they create propulsion that way. So yeah. I think you can see it working. Yep, you definitely can. It's amazing. It's brilliant. Those are quite small. Those are the small microbes that I talked about earlier. They're quite difficult to identify, except for some that have very distinct features. This one does have. And let me pull the book out again once more. Let me try and track this down. It's gonna go out of the frame, but you know, whatever. At some point I'm gonna get a different webcam for the books, because this, I think it's annoying for you and for me. But yeah. So let me see. It belongs in the clade hypotrachea, or however you want to say it. It's not very elongated, so yeah. This is a euplotus species. Those are euplotus species. Might be Euplotus affinis, but it's not that elongated. It might be Euplotus patella, sorry, not patella, patella. because it has, but it doesn't. This one doesn't have ridges, but yeah. Um, but it's. I'm pretty sure it's euplotis. It has that distinct shape. Or it may be Spidisca, but I'm really not sure. I'm pretty sure it's euplotis. Let me try and find it once more. By the way, this is a dead worm. You can see on the screen. On the bottom right screen. So yeah. Doesn't have a collar though. Collar of Siri. It's like this thing. You cannot see it very well. This thing. Anyways, very nice. Let me fix the screen again. The camera, by the way, has warmed up quite a bit. Anyways, let's continue. This is again a very large bubble. It has grown again. Needs water. Once more. Another worm. I think it's the same species, but different diet. Let's check it out. I found another thing, but I'm very curious about the worm, so you're gonna have to bear with me. Yeah, I'm pretty sure it's maybe a different species. I'm not sure. There's this junction inside of it. Not sure how well you can see it, but I can on the screen, so you might be able to, right in the middle of the screen, approximately right now. So yeah, might be different species. This is another microbe. I'm pretty sure about the genus too, because you can see the mouth. Not sure how well you can see it, but it's like a structure with some ridges that are parallel to each other, like a cone. I think you can see it right now. Sort of. Yeah, on the front of the microbe, like there's a hole that has some ridges. You can barely see them. And I think, let me check it out. from Tonya, possibly, but it's gone away again, but not so sure. Anyways, I'm not going to waste that much time today identifying, well, not waste, spend that much time identifying today microbes, but it's quite interesting if I'm going to create a better system for you to be able to watch more clearly what I'm seeing in the book. So yeah. So this that you cannot see yet. Come on. This in the middle of the screen. I'm pretty sure it's a dead rotifer. I'm pretty sure you cannot see it as well as I can, but I'm sorry about that. I'm going to zoom in so you can see it better. Go down. There we go. So let me close. There we go. Now you can see actually. That's impressive. That you can actually see. Good. So, this is a dead rotifer. Let's zoom in again. And it's a quite large one. I don't know that much about them, but they are animals. They're micro animals. And they're called rotifers because in Greek they're called wheel bearers. And rotifers, it's a similar thing, it's rotation. So yeah, it's because most of them have these structures, these arm-like things called the coronae. And these coronae are essentially like crowns, but those crowns are made of cilia, but different kind of cilia than the ciliates. It's confusing, I know, but you know. And they use them also to create vortices and pull detritus and particles around them and feed themselves with. But not just particles. They eat other microbes. They ciliates, flagellates, and other microbes also. Some of them do not even have those structures, those arm-like things. Some of them are just predators and eat other microanimals or other microbes, or microbes, not other microbes, because, well, it depends on what you define as a microbe, but whatever, yeah. So this behind, on the behind, this is the foot. It uses the foot to attach on stuff, and while it uses its cilia to create those vortices that would otherwise make it move. And yeah, that's quite a thing. Also, some of them, or many of them, use the foot to move, to jump around, sort of, because they're like in the water. But yeah, you know. Let's see if we can see anything else that I can identify. The mouth parts are like sort of in the middle, but yeah, I think this structure in the middle is the mouth parts that grab and tear down the prey, or the food anyways. Let me readjust once more. At some point I'm going to buy an adapter for the DSLR and put it there, and so I don't have to worry about this kind of stuff. But they're expensive. So yeah. I think I found another rotifer. This thing. I think a dead one also. Not sure why they're all dead, but yeah. Yeah, it's also dead rotifer. Yeah. I think maybe because... And my DSLR just closed perfectly. I think it has overheated. It's pretty hot. God damn it. Or it's out of battery. Out of battery. So let me change the battery and I will be back. Oh, there we go. It needed some resetting. But we are back. We are back, and now it's autofocusing. Great. Perfectly. I am very happy. Also, I found out that my card, that right now it's quite cool, was so warm. Next time I'm just going to take it out. Because I'm not recording anything. Like, the video is just being recorded on my computer, which is also quite hot, but not that hot. So yeah, let's get back to it. I left you with like a paramecium over here. It's quite a slow one, so I'm not sure. Oh no. Great news, guys. It is not a paramecium. Amazing. So, what is it then? I am not sure. It doesn't have the common structures of a paramecium, and it's quite slow moving. and it's very transparent. Like I was hoping that we could possibly see I'm hoping that we can. This is the rotifer again. That we can see the nucleus, but nuclei are usually pretty invisible. I've seen very few of them, and one was on a dead Vorticella, actually. So yeah, you can see all the vacuoles and stuff inside it moving. It's like jello. Well, it's quite similar. You can see the mouth right on the bottom. Now, on the bottom of the screen as well. It's rotating. Not sure why, but yeah. Another dead microbe. It's definitely a ciliate. I think it's another species of Paramecium, but I'm not quite sure. Probably not. Also, you might see that, you might notice that microorganisms are usually banded together. That's because different microbes prefer different micro conditions, different temperatures, different element concentrations, and in the slide that those differences exist. Between the one side of the slide and the other, there may be like so small of a difference that we will not notice, but the microbes surely will. Let me find it again, if I can, because I've lost it. I have officially lost it. Good, great, brilliant. Found it. So yeah, it's quite fun. I hope it's fun for you as well. That's a fast one, small one, but fast. I also think it's beautiful. For some, it may be scary, and well, I guess fair enough. But for me, it's just fascinating because, like, something you can never see without a microscope. Something so alien, so not close to us, and yet that's our cousins. Well, very far away cousins, but still cousins. You know. And yeah, it is. Love the Bob Ross emoji. Right now, you may be able to see the bases of the cilia. Yeah, you can. All those texture, like all this texture on the microbe, it's the basis of the cilia. There are so many of them, and there's very, very small, very small. So yeah. In length, I think they're close to maybe 4 micrometers. And in width, maybe 1 or 2, like 1 or 2, well, yeah, something like that. But they're definitely very small. Right now you just saw the vacuole that's responsible for the water expelling. Expel water. There it is. It's gonna do it again in a bit. There it is. Off again. Out with the water. I will find the name of that vacuole because it has a specific name, but I have forgotten it. And this microbe has two of them. Maybe paramecium, but again, not sure. By the way, we are closing the 2 hour mark, and I think that, god damn it, I am going to be interrupted again by another possibly dead rotifer. Rotifers are quite active, so yeah, that's a dead one. God damn it. And it has been broken down also. But this one is also attached to the slide. Oh no. I think this large part is the stomach. This large sort of vacuole shape bag, or however you want to call it, you know. So yeah, what I was saying before the rotifer interrupted me is that we are closing at the 2 hour mark, so in like 10-15 minutes I'm going to call it off because I'm very tired, and tomorrow I'm off to work at like 9 a.m. as usual. So yeah, you can join on Friday at around same time. So yeah. Oh, I'm not sure if the distilled water did any good. Because I'm seeing a lot of death right here. Next time, this is the first time I'm adding distilled water. So yeah. For you it's quite bright. I have to find a way to adjust that without much strife for you. Like, so it seems like the gods of microscopy want me today because, first of all, this is a dead microbe. Like, see how it's so broken down. But also, what I saw before I zoomed in was a small, well, baby Vorticella. So this is a baby Vorticella, or well, a motile Vorticella, because they have I think 4 life stages, and yeah, one of them is motile, so they can move around, because generally Vorticellae do not move, they're stationary, they cannot move from their place, but sometimes they just dissolve parts of their body or a part of their body and just move around. So yeah, this is a young Vorticella, or its model state. Those are somatic cilia that Vorticella generally do not have in their adult form. Well, I say adult, but you know. Also, for some reason, I think we can see the nucleus. It's very, very, very invisible, but very not invisible, but it's barely visible. But I think you can see it. Just a squiggly structure inside of the cell. Brilliant. Amazing. Vorticella have a very particular shape of nucleus. It's like an elongated. It looks actually like the large intestine. It has that kind of shape. Also, this movement that they do and that they get fatter and shorter, it's for protection. Normally, they have a stalk that they use to attach themselves onto sediment or the slide or some plants or water plants, obviously, but yeah, and so they can live their lives. But this one is trying to find a better place to live, or a place to live, so to say. So I'm gonna do a last search and then I'm gonna call it off because I am very tired and I need sleep, as I think we all do. So yeah, let me readjust the brightness and search. Because you know, you never know what you might find, and we find something interesting, it's gonna be nice. As a last treat, again the large microbe. Usually microbes are next to detritus because that's where food is. But right now, here everything is mostly dead. Again, a nematode. That's something interesting. Yep. Death and destruction. This is a dying microbe. What a treat. What a treat. I think the worm has busted it off and like all its cytoplasm is pouring out, but the membrane is still holding mostly. You can see the organelles around it. It's amazing. And it keeps. Yep, the worm is eating it off. And now it's busted. Amazing. Quite a meal. Quite a meal. Well, that's it from me. Let me change the view. That's it from me. Thank you very much for watching. And next time I will be a bit more prepared, and maybe I will have a better sample with more diversity because like this sample is a bit standard what you find. Nematodes, Paramecia, and generally detritivores. But again, thank you for watching and cheers. Have a nice evening or whatever part of the day you're on. Bye bye. See you in Friday.